I'm not sure. I know she has a 7:45, so they might just be running late.
Okay.
So we started with the early mammalian development, and we talked about how the biggest problem with the study today is that it's the smallest. It's really small, and the embryo develops in another organism, which also is hurdle, probably the biggest one to study, biggest hurdle. And then in vitro culture, up to the blastophore creation, the egg gets implanted in the female, so it's also hard to study. And so what we know is mostly from mice. And so the fertilization takes place in the ampulla region, and the cleavage in mammals is the slowest in the animal kingdom. It's 12 to 25 years apart. The first cleavage is meridional, and then it forms two loosely attached cells. And then second cleavage is one cell divides meridionally, and then the other divides equatorially, and it's rotational cleavage.
Okay, then what happens?
Then we moved on to parental chromosome changes. There's 3 that we talked about. So histones are exchanged with new histones, gametes, specific DNA methylation is removed, and then new methylation for totipotency and pluripotency is stabilized. So then we talked about by the 16th cell stage, each of the cells is hypomethylated, which means it's pluripotent. And then we talked about compaction. So in the 8 cell stage, blastomeres are arranged loosely. And then following the 3rd division, blastomeres change behavior and start expressing cell adhesion proteins, E-cadherin, on their surface. And then when E-cadherin is expressed, they start to come together and compact. And then you can't tell the cells apart. And then the cells divide into a 16-cell embryo called a mortar.
Okay.
So at the 8-cell stage, they're totipotent. When they divide into 16-cell embryo, the morula, they're pluripotent. They then undergo a change, and the inner cells are small and the outer cells are very large. The outer cells form the trophoectoderm, and tropho comes from trophic factor, which is a vital factor from the mother to form connection with the mother. And then the inner cells change into inner cell mass, and then those are called the blastocysts, which is like a 16+ stage. Trophoectoderm or trophoblast forms the non-embryonic portion of the embryo and forms the chorion, which is tissue to be embryo divide part of placenta. And they get O2 and nourishment from the mother. They also secrete hormones cause the mother's uterus to keep the embryo by modifying the mother's immune system to not reject it.
Okay, then.
So the first division from the 18 to 16 for differentiation, the generated cells, those are going to be the chorion, which will not be part of the developing embryo, and then the formation of the developing cells that will develop into the fetus. The inner cell mass cannot form trophoectoderm, but will form ovary cell type, and are pluripotent, and the 8 cells are totipotent with the trophoectoderm. Only tropho cells, so they lost differentiation. And then we talked about why to become the trophoblast or inner cell mass. And the decision to become the trophoblast or inner cell mass is the first binary decision of mammalian life. Pluripotency is governed by 3 key transcription factors: OCT4, SOX2, and NANOG. They can bind to their own genes and enhance each other to activate each other. So SOX2 and OCT4 form a dimer that activate NANOG, and the NANOG can bind to itself and produce more. And these activate, and then all 3 activate embryonic stem cell genes.
Okay. Okay. So what is happening that we have OCT4, SOX2, and NANOG. Okay. So what these are doing, they are actually recruiting RNA polymerase 2 to the genes required for pluripotency. And they are recruiting methyl transferase to the genes for differentiation. So if they are doing this, that means these genes are getting deactivated, or I will say, or repressed. And these genes, these are activated. Okay? So now, this is the cells which are going to be the inner cell, mast cell, right? Now, to be trophoblast, what we should do? To be trophoblast, now these cells are the cells which are outside, right? So to be trophoblast, what we should do, we should decrease the expression of OCT4, SOX2, and nano. You should decrease their expression. Now, that means you should do that thing, that the first thing you should do. And the second thing you should do is, you should increase or start the expression of the genes for trophoblast specification. You should do those things. To do things you should do. Now, so what that is performed by? That is, this whole activity is performed by CDX2. Now, CDX2 is a transcription factor. It's a transcription factor. Remember, transcription factors are like the proteins, but these proteins are unique. For some genes, they can act as a repressing protein also. They can bind to repressors and stop the transcription of those genes. And for some genes, they can bind to activating sequences and start the activation of that gene. So what this CDX2 does, it does two things. Number one, it acts as inhibitor. So when I'm saying inhibitor, so that means it acts as a repressor for the transcription of OCT4 and SOX2. Remember what OCT4 and also it does for the nano as well. Nano. But mainly it works on these two, OCT4 and nano. SOX2 is indirect. Remember we said that in the previous, we were saying that SOX2 and OCT4, they form dimer. When they form dimer, they go to nano and activate the nano, right? And then we said nano itself can activate nano, right? That was the flow chart we made last time. So if it is inhibiting OCT4, OCT4 will not be able to form dimer with the SOX2, right? So that means nano will not be activated. Whatever nano is there, if try to self-activate it, it's inhibiting nano as well as. So indirectly, SOX2 will also be inhibited. Making sense? Now, what it does, that's the first thing it does. So it means CDX2, it blocks OCT4 and nano, thus SOX2. And the second thing what it does, since it's transcription factor, it's going to the other gene. So it's going to activate the genes which are specific for the trophoblast specification. Those are EOMs, HAND1, EOMs, HAND1, and PSX1. PSX1. And those genes are specific for trophoblast specification. So what we are saying? We are saying that to be a trophoblast, or to be a cell of inner cell mass, that depends upon the type of genes which are going to be expressed there. And those type of genes are controlled by the transcription factor. We said if you have OCT4 SOX2 nano, what you're going to do, you're going to recruit RNA polymerase 2 to the genes. Those are required for maintaining the pluripotency. So that means those genes will be activated if you have RNA polymerase 2. Then what these do, OCT4 SOX2 nano, they recruit methyltransferase on those genes which are for differentiation. That means you're methylating those genes. If you're methylating those genes, you're inhibiting the transcription of those genes, right? So this means that this is happening in the inner cell mass. What is happening outside to be trophoblast? You should stop the expression of OCT4, SOX2, and nano. And we said that is achieved by the CDX2 because CDX2 is a transcription factor. What it does, that act as a repressor protein for transcription of OCT4 and nano. When OCT4 is decreased, it cannot form dimer with the SOX2, then cannot activate nano. There might be some nano present already. You need to inhibit that nano also because nano can activate nano itself, right? So ultimately, you've got both OCT4, nano, and SOX2 are repressed. Then what you need to do, you need to activate those genes which are required for the specification of the trophoblast cells. So CDX2 now acts as a transcription factor, as an activator for the gene EOMs, HAND1, and PSX1. Once it happens, those genes specify the cells to be trophoblast. Making sense? Okay, just talk to each other for a second and we'll go further.
Yeah, well, you just started off by saying to the gene required. So they want to be a trophoblast, so basically need to
Okay, anything that is doubtful? Okay, now. So we are saying that this is the first, now we just talked about the molecular players which are involved in making you a trophoblast or inner cell mass, right? But we said this is the first binary decision. So how the cell made this decision that you want to be trophoblast, you want to be inner cell mast cell. And that decision is essentially depend upon, like in the real estate, they say location, 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 right? That is exactly happening here. It's all location where those cells are. So what is happening to be a trophoblast? You need to lower OCT4, SOX2, and nano, right? And we are saying those are done by CDX2, right? So that means you need to have CDX2 upregulation somehow to lower down the pluripotency or the stem cell transcription factor. So what happened that this is the CDX2 gene. Just think about this is the CDX2 gene, right? On this CDX2 gene, you have a protein always attached to it called TEAD4. Now TEAD4 is there, but TEAD4 cannot do anything. What you need another protein called YAP. So another protein called YAP. This protein should come there. YAP should come. So YAP is there. YAP should come and bind here to the TEAD4. Once it binds TEAD4, it converts TEAD4 into the activator of the CDX2 gene. So YAP and TEAD4, when they are together, what they are going to do, they are going to activate CDX2 transcription. So CDX2 transcription is going to take place. So now, this should happen, right, to be a trophoblast. Now, think about what I told you, like the cells are like this. So you have some big cells here, some big cells here, and then there are cells here, right? We said these outer big cells are going to become the trophoectoderm. The inner cells are going to make the inner cell mass, right? So then, I also told you that factor, what you call location, is the critical thing here. So if you look at like kind of zoom in there, what you can see is, you can see here, for example, this cell is the outer cell. Right here. And then this is the another outer cell there. This is the another outer cell there. Okay. Then you have this cell which is a inner cell. Let me draw this. So this is the cell right here, is the cell that is inside. Okay. Now, when the cell are joining to other cell, there should be some cell adhesion protein there, right? And what are those proteins we did talk about, which are forming the compaction? What were those proteins? E-cadherin. So that means right here is a E-cadherin right here. Say this is another E-cadherin. Say this is another E-cadherin, another E-cadherin, E-cadherin, another E-cadherin, like this. Okay, so these are the E-cadherin. Okay. Now, here will be other cell also, right? Which will be surrounding this from here. There will be another cell like this. There will be another cell like this, right? Okay. So then what you will have? You have a E-cadherin here also, you have a E-cadherin here also, E-cadherin here also, E-cadherin here also, E-cadherin here also. So if you look here, that the cell which are, so this is the apical part of the cell, apical, and this is the basal, right? For this cell, this is the apical, and this is the basal, right? So the cells which are here, right here, they are surrounding from all the four sides by the E-cadherin. Okay? When they are surrounding from all the sides by E-cadherin, then a protein called so right there is a protein, that protein is right there, here, and right here. So this protein called AMOT. This AMOT protein is activated. This AMOT is activated. When this AMOT is activated, it goes down and it activates another protein called HIPPO. Activate another protein called HIPPO. Okay. Now this HIPPO protein, what it does, once it is activated, it activates another protein. That protein is called YAP. Now, when I'm saying activating, I should not use the term activate. I should use the term in different way. Activate means it phosphorylates this protein. Usually, I'm always saying phosphorylation. I use the term phosphorylate. It phosphorylates YAP. It phosphorylates YAP. So YAP has a lot of phosphate group attached to this. So this is the phosphate attached there. Okay. When the phosphate is there, what happens? This YAP is now signal for degradation. Okay. So phosphorylate YAP and leads to degradation of YAP. Now YAP is degraded. So what was the gene here? This is your CDX2 gene, right? Here is TEAD4. Here is TEAD4. Now YAP is degraded. YAP cannot bind to it. So CDX gene will not be activated. No activation there. Right? So it will keep on having the OCT4, SOX2, and nano. Making sense? Now what is happening here on the apical side? Right here, you will not see any cadherin because there is no other cell there. Right? So there are proteins, what we call, it's a group of protein, what we call partition defective proteins. Those are called PAR proteins. It's a whole family of PAR protein. Now, these proteins are critical in the development. even in maintaining these are also called polarity proteins. These are also called polarity proteins. So what does mean? So like we did talk about that first cell divide. We said when the stem cell divide, they have asymmetric division. Remember we did talk about, like we said, type A spermatogonial cell divide, give another type A spermatogonial cell, but then divide asymmetrically, you get one type A and type A1, right? So when you say asymmetric division, it means both the daughter cells, they are not going to get the same component. That would happen in the embryonic division also, right? We said the first division, you have the vegetae all on the vegetal pole. There is no vegetae on the animal pole, right? And then we said another BMP inhibitor, that is Norrin, that is usually on the animal pole. So that's called asymmetric division. That means some proteins, they are distributed asymmetrically. And that asymmetry is done by a special protein called polarity protein or partition defective protein. So they do that. So even if you look at, for example, a neuron, right? We have seen the neuron. Neuron have a long axon and a cell body. Now, why only one axon? Why not 2 axon, 3 axon, 4 axon? And why an axon and a cell body? That is determined by the polarity proteins. So polarity protein define how the cell will even look like. So there are these protein called partition defective protein. And then there are these proteins are usually localized on one side. In this particular case, those partition protein, a particular protein, partition defective protein in the FAR family, a protein is called PAC or PAC protein. Okay, that PAC protein is localized right here. So this is your PAC, right? And then there are what we call atypical protein kinase C. We write it APKC. Okay, so that APKC is also there. So that APKC come here. That is APKC. Location, right? So these are only on the apical part of the cell, which has no other cell to contact to it. That's how these proteins are localized there. Now what these proteins do, these proteins actually recruit a mode. So they recruit a mode with them. So now the a mode is there. A mode is there. When a mode is there, attached to them instead of cadherin, here a mode is attached to cadherin, right? So here a mode cannot be activated. Okay, so now a mode cannot be activated. When a mode cannot be activated, that means coming here, hippo not activated, right? If hippo not activated, then YAP not degraded. If YAP cannot degrade it, that means right here is the CDX2 gene. So TEAD4 is there. Now the YAP is there. Then that causes activation of CDX transcription, and then you have CDX2 there, and then CDX2 is going to activate HAND1, PXX1, and EOMs, all those three genes will be activated, and these cells are going to become profectinol. Making sense? Okay, so let's go over it. We are saying that this is the first binary decision. That decision depends upon the location. What is the location? And we are saying to be a trophoblast cell, what you need to do is you need to have CDX2 gene activated, because CDX gene is going to activate as a repressor for OCT4, SOX2, and nano, and will act as an activator for EOMs, PXX, and HAND1, right? If those transcription factors are activated, you are going to get change into trophoblast because you are going to inhibit the pluripotency gene. Now, that CDX2 gene can be activated by the binding of YAP to TEAD4. So YAP is a co-activator. When it binds to TEAD4, this whole gene is activated. Okay? So we are saying what happens in this is still morula, right? Still morula. So what happened, we said in the morula, at this stage, outer cell decide that they want to be profectinol. So now the location is such that the cells which are in the center, those cells have E-cadherin connection to surrounding cells. And when that happens, this C-mode protein, which is now bound to cadherin, gets activated. When that C-mode activated, that activates Hippo, and Hippo is a kinase. That kinase phosphorylates YAP protein. Once YAP is phosphorylated, YAP is destined to be degradation. YAP is degraded, and then there is no co-activator for TEAD4, so CDX2 gene is not activated. So no CDX2, that means you will have continuous production of SOX2, OCT4, and NAM. Whereas in the cells which are on the periphery, the apical side of this cell has no N-cadherin attachment. There what happened, the partition defective protein, PAC, start accumulating on the apical area, and that PAC is partnered by atypical PKC. Atypical PKC and PAC together recruit A mode, but once the A mode is there, A mode cannot be activated. If A mode is not activated, Hippo cannot be activated. If Hippo is not activated, YAP will not be phosphorylated. If YAP is not phosphorylated, YAP is going to go and bind to the TEAD4 and activate as a co-activator for the CDX2 gene, and CDX2 will be then expressed here. That is going to lead to expression of PXX, EOMs, and HAD1, and then these are going to change into the trophoblast cell. Making sense? Any doubt? Okay, they talk to each other for a second.
All I know is that the activating CDX gene means that there's decreased levels of OCT4 or SOX2 nano, which happens in the outer cells. And if it's CDX is not active, then OCT4 or SOX2 nano are not increased. CDX used to be upregulated somehow in The inner cells, when it's completely surrounded by E cadherin, AMOT is activated, which activates Hippo and phosphorylates ER, which leads to degradation of, which inactivates the CDX gene. So inner cells have an increase in OCT4, soft now. And then position effective proteins, PAR, label proteins, localize on one side, so the outer cells, and then our examples called a PAC protein, and that and, what usage do these stand for? Yeah, okay, and the POC, recruits AMOT, which usually detected to hey, since it's recruited, there is no connection. So it can't be activated, which inactivates Hippo. YAP is not degraded, so it can attach to CDX gene and activate that, which leads to increase or decrease the outer cells further.
Any doubt?
Quick question, the apical and basal side applies to each individual cell, right?
That is for each individual cell, but the apical surface is the top surface, basal surface is the lower surface of the cell. Yeah. But here in the periphery cell, there is no cell above the apical surface here. So there is no cadherin attached to any cadherin from the other cell. So that's how the distribution of these PAR protein is going to take place. That's kind of telling, hey, I'm going to be on the apical side, which doesn't have any cadherin attached to the cell. Any other question, doubt? Okay, now. So, we are going to talk about the next thing, what we call cavitation. Process. So what happened that we are still in the morula, right? So in the morula, what is happening, the cell division is continued. Cell division is taking place continuously. By the 64 cell stage, so morula start at 16 cell. Remember the 8 cell divide, 16 cell, and then they huddle, right? So they're dividing, so by the 64 cell stage, (13 cells. So these 13 cells, which were like at the right location where there was no activation of CDX2, right? So these 13 cells, they said, let us become the ICM cell. And trophoblasts are separated into separate layers. So what does it mean? It means neither ICM cell or neither trophoblast cell, right? So neither of which out of these two contribute to each other. Neither of which contribute to each other. Now, ICM cells secrete factors to support the development of trophoblasts. So what is happening there? You are having the outer cells right here. These outer cells are there. So they are also growing. They are now dividing separately. And the cells inside, they are also dividing separately. So neither of it's contributing to each other, right? They are dividing. So these are now, so these are the ICM cells, and these are the trophoblast cell. Okay, now what happens? Now they need to go to the next stage. So what happened that these trophoblast cell, what they do, so this is still a morula. This is still a morula, right? So trophoblast cells, what they do, they secrete fluid into the morula. And what they do. So the membranes of these trophoblast cells, which are outside, they do have right here, every cell, they do have sodium-potassium pump and sodium proton pumps. So what they do, these are all active transport, right? So what they do, they start secreting from inside, from here, they start secreting here, and that is mostly on the apical side of those, which is the apical side. They are more concentration on the apical side. They are less on this side. Mainly they are concentrated on the apical side. What you can say is like animal pole type, whatever you want to say, right? So from here, what they do, they start secreting sodium ion here, sodium ion here, a lot of sodium ion here, a lot of sodium ion come here. So now what is happening? When you're having a lot of sodium ion there, that will be followed by the water, right? So what happened? That sodium ion come there, with that water come there, and that leads to formation of a, so these are your trophoblast cell. They secrete sodium here, sodium, lot of sodium. When they secrete a lot of sodium, that is followed by the water. So water come osmotically, because of the osmosis, right? Because you have more salt here, that should be followed by the water, right? So then what happened? On this side, you get a lot of water start coming here. And that water pushes these cells on one side. And that leads to formation of all these inner cell mass cells are kind of like this. Okay, so now these are the ICM cell. Right here is the water-filled cavity, right? That water-filled cavity is called, what you call it in blastula? Blastocele, right? You have blastocele, and now this structure is called blastocyst. And remember that this blastocyst or morula, they still have this zona pellucida surrounding them. So this zona pellucida is still surrounding them. And these ICM cells, these ICM cells are actually called embryonic stem cells. So ICM cells, inner cell mass cells, are called embryonic stem cell. These are the cells with, by the name the whole world get crazy. Alright? So these are the embryonic stem cells. And the cells surrounding are trophoectoderm. So embryonic stem cells, they are going to contribute. So embryonic stem cells, they are going to contribute the new embryo. So all the body system are going to be made from these embryonic stem cells. Trophoectoderm, they are going to give extra embryonic tissue, what we are going to call as placenta, or the placenta, the chorion part of the placenta. We'll talk about how that is formed. So this thing is clear. So what we said, that these trophoectoderm cells, they have, so they keep on, morula keep on dividing, reaches up to 64 cell stage. When that happens, so cell separation occurs. So central 13 cells say, hey, we are ICM, you are trophoectoderm, we don't contribute to each other. They keep on dividing. ICM divide, trophoectoderm divide. So ICM cell, they secrete some factors, those factors continuously support the growth of the trophoblast cell. And then the trophoblast cell, they start acquiring sodium potassium pump and sodium proton pump, and they start secreting sodium inside the morula. And when a lot of salt concentration come here, a lot of sodium come here, that is followed by the water. Water comes osmotically, and then water starts secreting here, and water starts filling it, and these cells are now pushed to one side. So the cells which are pushed to one side, these are inner cell mass, ICM cell, and these cells are embryonic stem cell, going to give you embryo proper. And the cells which are surrounding it are trophoblast cell. These are going to give you the chorion, and we are still saying that all these are surrounded by, still by the zona pellucida. Making sense? Okay, now talk to each other, then we'll go further.
Cavitation, like cavity.
And the rest, only has all the trophoblasts, right? These 13 cells are synthetically and then they the ICF5 is there cells because they just get pushed down. I don't know why. And that's they don't talk about how it drops.
Makes sense though. I think,
Confusing?
no.
Oh, you guys are okay? You're fine? Okay, now. So this is your so this is your ovary right here. This is your oviduct. These are temporary, so this is the ampulla region. So here is your, this is your zona pellucida, and this is the oviduct. Right here is the uterus. So, uterus is the place where the implantation is going to take place. right? So this development is taking place in this thing. Everything is happening zona pellucida. You had like fertilization changed to morula, morula changed to blastocyst, right? We don't call it blastula here, we call it blastocyst. And then this is moving here, and it's supposed to reach here. And here it need to implant in the uterus, right? So what is happening, what we're going to talk here is, we are going to talk about escape from zona pellucida. And actually that is hatching. So we usually say the birds hatch or the reptile hatch from the egg. We also actually hatch from the egg. So we hatch inside the other body, so people don't know we also hatched out of eggs. So what happened? This zona pellucida is actually critical, because where the implantation should take place, that is the uterus. Okay. So this zona pellucida, what it does, zona pellucida protects the embryo from implanting in the oviduct. Otherwise, the cells are very sticky. They can stick anywhere, right? So what is happening? So they don't allow embryo from the oviduct. So what is happening here? Right here is your zona pellucida. This is your ZP. And here are your trophoectoderm cell. These are your ICM cells. So this is your ZP, ICM, and your trophoectoderm cell, your trophoblast, and write trophoblast here. Okay, so what happened that this zona pellucida is preventing adhering of this embryo, which is developing here, to the oviduct. Right? And it need to implant here. Think about, so how it comes out. It comes out that these cells which are right here, the trophoblast cell, how you break the other cell? Trypsin, right? So these cells actually start secreting trypsin here. So these cells, they start secreting trypsin here. Trypsin secretion. And what trypsin does? Trypsin secretion is going to kind of digest here. Let me draw it further. So what is going to happen? You have the trypsin secretion there. So trypsin secretion is going to digest. So trypsin digests ZP and form a hole. Right? It form a hole, and what happened? This embryo, which is developing here, this is your embryo. This embryo squeeze out. From zona pellucida. So just squeeze out. Okay. And this squeezing out is actually called hatching. So hatching takes place, so now this embryo is out. Blastocyst out of zona pellucida. Now this is actually very important, very critical thing. that zona pellucida is surrounding it. So if the zona pellucida, if this process occurs, now this process is well timed, that this hatching only takes place in the uterus. If this hatching takes place somewhere in the oviduct, that lead to very dangerous conditions. So hatching in oviduct is dangerous. It's dangerous for mother. You might have heard of this term called ectopic or tubal pregnancy. That is what is, that this hatching takes place in the oviduct. And when that hatching takes place in the oviduct, or this fallopian tube, people call it in common language, then this embryo will start implanting here in the oviduct. It's supposed to implant in the uterus. And that condition is going to lead to severe bleeding. And that might lead to death and so many things, so many complications there. Okay. So trophoblast cell, they secrete trypsin only when they are in the uterus. But sometimes this mechanism go every, and when that happens, at that time, you get tubal or ectopic pregnancy, and that's a dangerous condition. So far it's okay? Now what happens? So hatching has taken place, and from hatching, you need to have implantation. Hatching to implantation. Okay. Now, once outside the ZP, outside the zona pellucida, right? Now, blastocyst makes direct contact with the cells of uterus, and those cells are called endometrium. And when you say blastocyst, what cells are outside? Outside are trophoblast cells, right? So now these are the cells of, now this is, say, the surface. So these are the surface, these are the endometrium of the uterus. Now what happened? When the ovulation takes place, right, we said corpus luteum is formed. And we said corpus luteum secretes a lot of progesterone, right? And what was the purpose? To prepare uterus for the coming embryo. So what happened? So progesterone and estrogen also from corpus luteum. What that does, that tells these cells, which are endometrium cell, to make, right here, a lot of things are secreted out. So what are those? It tells them to secrete sugars. Remember, sugars are sticky, right? And then also tell these cells to make some cell adhesion proteins on the surface. Those are fibronectin and then cadherins. Cadherin. So those are secreted there. And then you have this embryo coming out here. Now this is your embryo right here. Or I will say blastocyst. Right? In this blastocyst you have these these are the ICM cells right there. Right? So right there what is happening when you are having this, these cells are secreting blastocyst is there, and blastocyst is coming there, endometrium has secreted sugar, fibronectin, and cadherin. So you need something to stick there. And that sticky thing is produced by the trophoectoderm cells here. These trophoectoderm cells, they start also making some cell adhesion protein. And what these do, so they secrete integrins. Now integrins are there. Integrin allow them to attach to fibronectin, cadherin, and sugar. So this is kind of stuck there. Right? Once it is stuck there, so attachment. So first step is attachment to endometrium. And we know that attachment took place because the endometrium was prepared by the hormone progesterone and estrogen. They start secreting fibronectin, cadherin, and sugars. Then trophoblasts secrete integrin, and that causes attachment of this early embryo with the uterus. Now what happened? Second, this trophoblast secrete proteases and digest extracellular matrix of endometrium. So, what is happening? It starts secreting proteases, these cells. So these proteases are the enzyme are coming there. What they do, they start digesting these cells like this. And when that happens, they start digesting like this, and this embryo or blastocyst, still blastocyst, remember, it actually starts burying itself in uterine walls. So it starts burying itself in uterine tissue. Okay? So attachment is there, starts secreting proteases, these proteases start digesting extracellular matrix, and it starts burying inside. Now, this process of coming here, burying inside, is called implantation. So the implantation has taken place. Most likely, it happened 5 to 9 days after fertilization. Usually day 9. Okay. Now the positive test for pregnancy is not till yet. That comes 3 to 4 days after implantation. And that's not a common test. That test is by the special hormones, what we call human chorionic gonadotropin hormone. Human chorionic gonadotropin hormone. So that hormone is tested in the blood. Think about all the hue and cry which people are making about embryonic stem cells. Once it implanted, it is no more blastocyst, it will immediately start the gastrulation, we'll talk in the next lecture. During budding process that starts. No one will know that a person is pregnant. The size of this blastocyst is a period on your book, 100 micron, remember we said. It's dividing, but it's not growing that much because surrounded by zona pellucida. Is there any way to find this out? When there is no test even telling that a person is pregnant. There is no way to locate this thing in the whole tube, which is that room. So all that myth which people have created, like these are human embryonic stem cells, and these are human embryonic stem cells only at this stage. After this, there will no more be human embryonic stem cell once they implant it. So there is no way abortion can produce human embryonic stem cell. So what we are saying? We are saying escape from zona pellucida take place because these cells, and zona pellucida is required because you don't want it to attach to the oviduct. You want it to attach to the uterus. And that zona pellucida, when the time comes, when the egg reaches, not the egg, the developing embryo reaches the uterus, the trophoblast cell, they start secreting proteases that digest zona pellucida and the whole embryo squeeze out. And we call that process hatching. The hatching takes place. Once that hatching takes place, we know that the endometrium or the cell lining of uterus are already prepared by the corpus luteum, progesterone, and estrogen. Under that influence, these cells, they start secreting fibronectins, sugars, and cadherin, attachment protein. And then trophoblast, they start secreting the integrin, and that causes the attachment. Once that attachment is taking place, then what happens that these trophoblast cells, they start secreting proteases. Those proteases are going to digest the extracellular matrix, and this developing embryo will bury itself inside the uterine wall or uterine tissue. That is called implantation. And we said that this happening usually at day 9. And the best of the best tests, they basically cannot detect the pregnancy by the 12 days. And they use a hormone in the blood. It's a very sensitive test. They test human chorionic gonadotropin hormone. So then we are saying that there is no way at all by which you can detect this embryo in the uterus and do the abortion to get these embryonic stem cells. Any question here? We can talk to each other, and then if there's any confusion, we can talk, and then we'll move on.